Welcome to video 2.6. In this video, I'll be covering the secondary structures of nucleic acids, as we've discussed the individual building blocks, the nucleic acids, the chains, the long molecules, DNA and RNA polymers, but there is one extra layer of complexity, which is the secondary structures that these can make. So again, starting with what we initially have worked with, the primary sequence of DNA. When we say, to understand secondary, we should talk about primary. That is the 5 to 3 direction and the bases we're using, right? So GCAT 3 prime, for example, right? We have the direction and we have the individual bases, right? That would be the primary sequence that we read for information. For RNA, it'd be very similar. Just remember that any T's turn into U's, right? So this is the primary sequence of RNA or DNA. Now, going forward from there, we should remember that the structure determines the function. So DNA and RNA polymers are similar. They have negative backbones, and they're long pieces of spaghetti, we can imagine. Maybe they can coil up. But what do secondary structures change? So again, a single molecule of DNA could be a long strand. Maybe it's coiled up on itself, whatever, right? Part of the reason I think it's nice to visualize long strands is because, again, DNA and RNA are negatively charged, right? Every base, every phosphate brings along with it a negative charge. So these things don't really want to coil up by themselves because negative charges repel each other, right? That's an important consideration. Now, multiple paired molecules, and this is kind of back to that idea of having a matching sequence, right? If I have a piece of DNA that is GCAT, and then another piece of DNA that matches up with it, so G's pair with C's, C's pair with G's, A's with T's, and T's with A's, this is now, we can view it as a larger molecule with secondary structure, right? These are two individual molecules. They're not one molecule all of a sudden, but the fact that they're connected gives it this new structure in space. And remember, structure determines function. New structure, new shape, new activity in the body. So these multiple paired things, right? RNA could also pair with itself, we could imagine, right? Or a piece of DNA can pair with a piece of RNA. So this secondary structure and the actual structures they make are something we need to understand in a little more detail. For DNA, this comes back to the classic double helix. So again, the Watson and Crick experiments, Rosalind Franklin as well, but two separate strands of DNA connected by hydrogen bonds and other forces, right? That's not the main one, but we talk about G's having 3 H bonds with C's and A's having 2 H bonds with T's, right? The beta form of DNA is the standard form, and I'm not going to dig into too much specific detail, but you'll notice a few different things, right? First and foremost is that there is a twist, right? So there's a twist to it, right? Here is one strand, so I'll color one strand in blue. We're going down this way around here, right? And the other strand will be in purple. You can see how it goes the other way. So we make this helix-like shape. Now, if you look at the actual spaces in between, you'll see that there's areas that are large, and then areas that are small, and then areas that are large, and then areas that are small, right? These are kind of gaps in between the backbone. Keep in mind here that the orange stuff, right, all the orange stuff in here is DNA bases that are stacked up on top of each other with H bonding, really stuck together, right? They're attracted to each other across, keeping the strands together, and they're attracted to each other just by their van der Waals forces and other dipoles. So the DNA bases are in the middle, where the white part on the outside, blue and purple, this is your backbone, right? These are your two distinct backbones of the ribose sugar. So I'll even point that out here. You have the ribose sugar and the phosphates, right? So super negative, or that's a positive charge, super negatively charged, right? All across the outside here. And these grooves in between, right? They kind of give you access to the DNA bases in a little bit, in a bit of you can imagine it that way, right? There's a gap between the backbone. We call these big groups the major groove, or the major grooves, and these small gaps the minor groove. And you'll see they alternate. This could matter because some proteins, for example, might interact by binding into the major or the minor groove. Right? This is a feature of DNA So no matter what that DNA sequence is, you're always going to be seeing major and minor grooves. That's the beta helix. And if we want to view it, imagine it from the top-down view, we can imagine that there's this central core of DNA genetic material, right? And all around it, we have those, from the top down, we'd see basically just the outside of this is like a white kind of casing of all those phosphates. So all that important genetic information is kind of wound up, kept safe, and surrounded by a shield of negative charge to keep things away. Cool, right? In this state, this DNA is not accessible to be read. Again, helicase needs to unwind the helix to free up and expose those DNA bases. But this is a very stable way, right? The beta helix is a secondary structure of DNA. Now, we call this dsDNA for double-stranded. Right? Single-stranded DNA is just regular DNA by itself without that secondary structure. Right? What makes double-stranded different to single-stranded? A few couple things, right? Obviously, the structure when it comes to binding proteins, so proteins that want to bind DNA, the shape is totally different, right? The shape and the nature of that helix is going to be the minor and major grooves. That makes double-stranded DNA very different than single-stranded DNA, right? The other thing is how much H bonding there is, right? So effectively, there's not too much to really dig into here, right? But double-stranded DNA is much more tightly secured. Single-stranded DNA is less stable, right? Single-stranded DNA is exposed to the entire, like if you have a piece of spaghetti, it's exposed to water on all sides. Where when you have that kind of core DNA thing here, it's really protected. So without further ado, that's a quick little foray there. Let's understand one last concept here with the DNA helix, which is the twists. So sometimes you'll see these called right and left-handed. And it can be really hard to tell the difference because looking at these, it's not really clear to me that this is right and this is left, right? So how do you do it? One thing and one trick that I was taught, I think by a professor at some point, is to view it like a staircase, right? So the idea here is on this right-handed helix, you can see that on top of the black is going on top of the red, and then behind, and then on top, and then behind. Here, on top, behind, on top, behind. So what's the difference? Imagine that you're walking down a spiral staircase. So you always start at the top. And here is a spiral staircase. You're starting up here, and you're walking down. The question is, because this is also a helix, which direction are you turning? In this case, as we're walking down, we're going to turn to the left. Like if you're taking that first person view, turning to the right, you would hit the wall, right? You would need to constantly be turning to the left to walk down the spiral. Therefore, it is a left-handed helix. So going back up to the DNA, the right-handed helix, if you're to imagine walking down this black strand, on top and then behind, you'd be turning right. Same thing here for the left. On top and then behind, you'd be turning left. So imagine that analogy is a helpful way to be able to look at left versus right-handed helices, right? For DNA, let's take a look at this beta form, right? We can walk down this kind of staircase, so we're going down to the right. This is a right-handed helix. So another important part of this is that it's a right-handed helix, which again, you can memorize, but hopefully you can just look at the sequence of DNA now, imagine it like a staircase, and see how you're walking down to the right. All right, and that wraps up the end of secondary structure here. So for the last topic, we'll be diving into detecting DNA and RNA using different experimental techniques and the breaking of these double-stranded helices back into single-stranded and all those cool applications for that. So I'll see you in the last section, video 2.7, coming up.